Hello everyone, my name is Isaac Hayden, and today I will be presenting on G protein coupled receptor signaling, also known as GPCR signaling. Before we begin, I want you to think about how quickly the body can respond to a signal. Imagine that suddenly you hear a loud noise behind you. Within seconds, your heart rate increases, your breathing becomes faster, and your muscles prepare for you to react. Those changes begin when chemical signals such as epinephrine bind to receptors on surfaces of cells. Many of these receptors are GPCRs, and GPCRs are one of the most important receptors that are in a family in biochemistry because they allow cells to collect information outside the cell and translate the information into a response inside the cell. They participate in vision, smell, taste, things like that, hormone signaling, immune regulation, cardiovascular function, metabolism, and many other things. So during this presentation, I will explain what GPCRs are, how their 7 transmembrane structure support their function, how ligand binding activates, the G proteins, how second messengers amplify the signal, how the pathway changes cell behavior, and how the cell eventually, does the signal and shuts down and the signal is done. I will then connect this pathway to diseases and modern drug therapy. The main idea I would like you to keep in mind is that a GPCR acts as a molecular communication device. It receives an extracellular message, changes its shape, and then initiates an organized biochemical response outside the cell. So here are our learning objectives. So by the end of this presentation, you should be able to accomplish 4 objectives. First, you should be able to define GPCRs and identify the features that make them different from other membrane receptors. In particular, you should recognize the characteristic 7 transmembrane articulature. Second, you should be able to explain receptor activation. This includes ligand binding and conformational changes in the receptor, the exchange of GDP to GTP on the alpha subunit, and the activation of downstream effects proteins. Third, you should be able to describe the major second messenger pathways associated with Gs, GSs, GIs, and GQ proteins. We will focus on cyclic AMP protein kinase, a phosphatase C, IP3, DAG, calcium and protein kinase C. Fourth, you should be able to discuss why GPCR signaling matters clinically. We will go over some examples of this in our presentation. And as we move the presentation, notice that each objective builds on the one before it. We will begin with the receptor itself, move step by step through the signaling pathway, and finish with the physiological and clinical consequences. So here we are with the G protein. So what are G proteins, GPCRs? G protein coupled receptors, aka GPCR, are a large family of integral membrane proteins. The phase integral membrane protein means that the receptor is embedded within a phospholipid bilayer, so it's on the outside of the cell, as we can see here. So it'll be on the outside, and this is our phospholipid bilayer here. So it's there rather than loosely attached to one side of it. GPCRs are often described as a larger receptor family in humans. Although individual GPCRs recognize different signals, they share a common structure plan. Each receptor contains a 7-membrane spanning alpha helices. That is why GPCRs are also called 7 transmembrane receptors. The receptor is positioned so that part of the protein faces the extracellular, which we can see here, the extracellular space here on the outside environment, and part faces the cytoplasm, which is on this side, on the inside of the cell. This would be the inside of the cell here. The extracellular facing region interacts with ligands. A ligand is any molecule or stimulus that binds specifically to a receptor, influences activity, and GPCR ligands are remarkably diverse. They include small molecules such as epinephrine, dopamine, larger proteins like hormones and things like that, lipids. The receptor does not usually transport the ligand into the cell. Instead, ligand binding changes the receptor's conformation, which means once it binds to the ligand, it changes the shape of the protein rather than it going inside of the cell. That structure change is transmitted across the membrane and alters the intracellular surface of the receptor. On the cytoplasmic side, the activated receptor will then interact with the G protein, and that means the G protein contains the subunits, the alpha, beta, and gamma, and this is what at resting state these subunits are associated with one another, and the alpha subunit holds GDP. The name G protein comes from this ability to bind genuine nucleotides like GDP, GTP. And so GDP is associated with the inactive state, while GTP is associated with the active state. Therefore, the receptor does not directly carry out every cellular response. Instead, it activates the G protein, and the G protein will then relay the message in the intracellular enzymes or ion channels. This is a useful point to separate these levels of the pathways. The ligand is the first messenger because it carries information from outside of the cell. The receptor then detects the message. The G protein downstream affects the transmitting of the signal inside the cell. So then that's when we get our second messengers, which we will discuss later, and distribute and amplify the signal that was initially done by the ligand through the cytoplasm. And GPCRs are therefore key regulators to cell communication. They allow cells to respond rapidly, selectively to changes in the environment. A liver cell can respond to glucagon, a heart cell can respond to epinephrine, a neuron can respond to a neurotransmitter, because each of these cells express a particular combination of receptors and signal proteins. So moving on, now that we have defined GPCRs, let's examine their structure more closely. The most recognizable feature here in the group of 7 alpha helices, so you can see the helices are here, the 7 of them, which they are labeled. These helices are numbered 1 through 7 in the diagram. Each helix is made up of amino acids arranged in an alpha helical structure, and many of the amino acids, the side chains exposed to the membrane, are hydrophobic. This hydrophobic character is essential because interior of the phospholipid bilayer is nonpolar. So hydrophobic side chains can interact favorably with the fatty acid tails of the membrane lipids. In contrast, the portions of the receptor exposed to the extracellular fluid or cytoplasm contain more hydrophilic and charged side chains, because those can interact with those according to their affinities. And the receptor also contains extracellular loops, intracellular loops, and extracellular amino terminus, and the intracellular carboxylic terminus. Depending on the GPCR, the ligand may bind within the pocket found by the transmembrane helices to the extracellular loops, or to a larger extracellular domain. The intracellular loop and carboxyl terminal region are especially important for the combination with G proteins and regulatory proteins. When a ligand stabilizes the active receptor conformation, the cytoplasmic end of the helices will then shift. This exposes or reshapes the G protein binding surface. In simpler terms, ligand binding on the side of the membrane changes the protein shape on the other side. So ligand binding here will change the shape in here. This is a form of allosteric regulation. The ligand does not have to touch the G protein directly. Instead, binding to the extracellular or transmembrane site changes the receptor conformation, and that conformational information is transmitted through the protein, the purple and blue areas here that are in the figure. Help to interpret that. They represent the 7 connected membrane spanning segments of the receptor in the polypeptide. Likewise, the G alpha, G beta, and G gamma, which are here, proteins shown below the membranes, are separate subunits that associate with the receptor only on the intracellular side. The structure also explains why GPCR can recognize so many different ligands while using a broadly similar activation mechanism. So the extracellular and ligand binding regions can vary, subsequently providing specificity. So the internal architecture and cytoplasmic signaling surfaces remain conserved enough to activate related families of G proteins. So the key structural, so it's key to the structure. So moving on to the next slide. So we'll move on to the activation mechanism here. So I'll kind of go through how you initially get here's the ligand, it binds here to the binding site, so now it's bound, and then everything under here in the intracellular starts to change conformation. It'll cause the conformational change because of the ligand binding, and then here we see the end result with the ligand bound and then the completed conformational change. So in a step 1, the ligand does bind to an initial inactive GPCR. Before the ligand binds to the receptor, it will fluctuate among different conformations, but the inactive site is generally favored by a particular ligand. The nearby heterotomeric G protein consists of the alpha, beta, and gamma subunits, and the GDP is bound to the alpha subunit, which you can see in the diagram. In step 2, the ligand binding stabilizes an active receptor conformation, The activated GPCR interacts with the G alpha unit and then acts functionally as a guanine nucleotide exchange. That's a factor, and this phase is important. The receptor does not phosphorylate the G protein, and it does not convert GDP chemically into GTP. Instead, it promotes the release of the GDP from the G alpha, and once the GDP leaves, GTP then binds to the GTP, is abundant in the cytoplasm, so the empty nucleotide binding pocket is quickly occupied by GTP. The exchange of GDP for GTP changes the shape and the activity of the alpha subunit. In step 3, the G alpha GTP becomes functionally activated and changes its interaction with the beta gamma complex. Many introductory diagrams will show this. So the G alpha complexity separating from G beta gamma. This is useful in this model, as we can see here. Although in living membranes, the subunit may remain close together and move dynamically, the key biochemical point is that both G alpha GTP and G beta gamma are now interacting with the downstream effects. An effect is a protein whose activity is altered by the activated G protein subunit, and common effectors include adenyl cyclase, phosphatase C, and ion channel, which affect the activated depends on the receptor and the G protein subunit of the cell. So we'll move on to the next slide. So now we're going to be talking about the second the second messengers. So for the second messengers, here we can see there's second messengers pretty much help to kind of amplify the initial signal that the ligand had caused by binding to the initial site from the initial ligand. So now we'll move on to the second messenger. And so in this, the next stage of the pathway is the second messengers. Second messengers are small intracellular molecules or ions that relay information from an activated receptor and affects the system through system to targets inside the cell. The first pathway of this side in the G pathway, the letter S refers to stimulation. When G alpha is activated, the stimulus adenyl cyclase. When it's activated by the stimulus adenyl cyclase. Adenyl cyclase is a membrane-associated enzyme that converts ATP into cyclic AMP or CAMP. So cyclic AMP then binds to and activates the protein kinase A, commonly abbreviated PKA. So protein kinase A phosphorylates specific serine and threonine residues on the target protein. These targets may include the metabolic enzyme, ion channels, or transportation regulating proteins. The result depends on the cell, its type. In one cell, PKA may increase glycogen breakdown. In another, it may alter, cardiac contraction. In another, like your cardiac heart cells. And it may change gene expression. The second pathway in the GI pathway, the letter I represents the inhibition. Activated G alpha I inhibits adenocyclase. As a result, the production of the cyclic AMP will then decrease. PK activity is then reduced, and the cellular effects driven by the pathway will then become weaker. It is important to recognize that GI does not simply mean that the entire cell becomes inactive. It means that the specific signaling branch, usually adenocyclase and CAMP production, is inhibited. At the same time, the beta gamma subunit released from the GI protein can then regulate ion channels or other effectors. The third pathway is the GQ pathway. Activated G alpha Q stimulus phosphatase C. Beta, usually abbreviated PLC beta, phosphatase C, acts on membrane phospholipid called phosphodialyl 4,5 on PIP2. So PLC will then cleave from PIP2 into 2-second messengers IP3 and DAG. IP3 is soluble and diffuses through the cytoplasm to the endoplasmic reticulum. There it binds to the IP3 gated calcium channel and causes calcium ions to be released into the cytosol. Calcium is itself an important second messenger. It can bind proteins such as clonium and influences enzymes, secretion, and muscle concentration and gene regulation. DAG, in contrast, remains within the cytoplasmic membrane because it is hydrophobic. DAG works together with calcium to activate protein kinase C, or PKC. PKC then phosphorylates target proteins and changes cellular activity. The bottom statement on the slide is especially important, so let's focus on that. Because different G proteins produce different second messengers, and different second messenger proteins produce different cellular responses. The responses does not determine the response by itself. The response depends on either signaling network present in the cell. Second messengers also create amplification. One activated effect enzyme can generate many second messengers and molecules. Those molecules can activate many kinases, and it goes on and on and on. However, amplification must be balanced by regulation. Cyclic AMP is broken down by the phosphodiester kinases. Calcium is pumped back into the storage components or out of the cell. IP3 and DAG are metabolized. Without these control mechanisms, the signal would persist and be lasting too long, which you don't want that. Okay. So we'll move on to cellular effects. After the receptor G protein effects and second messengers have been activated, the pathway produces changes in the cell behavior. This slide will highlight the 4 broad categories of how this works. So it will be metabolism, gene expression, secretion, and contraction. First, GPCR signaling regulates metabolism. For example, glucagon receptors in the liver activate a G pathway that increases cyclic AMP and protein kinase activity. This promotes glycogen breakdown and supports the release of glucose into our bloodstream. Adrenergic receptors can also regulate lipid and carbohydrate metabolism during stress or exercise. And so, second, GPCR signaling alters gene expression, where protein kinases activated by second messengers can enter the nucleus or phosphorylate transcription factors as well is a well-known example of the CREB, the cyclic AMP response, element binding protein. When phosphorylated, CREB can influence the transcription of specific genes. Therefore, a signal that begins at the plasma membrane can eventually alter which protein the cell produces. Another is the GPCRs regulate secretion in our endocrine cells, neurons, and other secretory cells. GPCR signals may change, calcium concentration, our protein kinase activity. Those changes can promote or inhibit the fusion of vesicles with the plasma membrane. This may be the release of hormones, neurotransmitter, digestive enzymes, or other signaling molecules. And last, GPCR pathways influence the contraction. So the GQ couple receptors can increase intracellular calcium, and calcium is a major regulator of smooth muscle concentration. This is relevant in blood vessels, respiratory tract, and the gastrointestinal system. Depending on the receptor subtype and tissue, GPCR activation can produce either contraction or relaxation. So, for example, epinephrine can increase heart rate through beta-1 adrenergic receptors and promote airway relaxation through beta-2 adrenergic receptors, and influence blood vessel tone through alpha adrenergic receptors. And so the ligand is the same in all these cases, but the receptor subtype and cellular context are different, which will then cause the different examples that I just said. So now, last, we'll talk about signal termination. A signal pathway must be able to turn on, but it also must be able to turn off. If GPCR signaling continued indefinitely, cells would become overstimulated, lose sensitivity, or experience harmful changes in metabolism, calcium concentration, and in gene expression. The first termination mechanism shown here in our example is GTP hydrolysis. The alpha subunit has initiate GTPase activity, which means that it can hydrolyze its bound GTP into GDP and inorganic phosphate. So we can see that here in our example. When GTP is converted to GDP, G alpha returns to the inactive conformation and stops regulating the effector. The GDP bound alpha subunit then reassociates with the beta gamma complex, which we can see that's going on here. This reforms the inactive G protein and prepares it for another cycle of activation, where another ligand would then have to come in and reactivate the whole process. The rate of GTP hydrolysis can be accelerated by regulatory proteins known as RGS proteins, are regulators of G protein signaling. These proteins help determine how long a G protein signal lasts. The second mechanism involves beta arrestin. After receptors have been active, GPCR kinase can phosphorylate sites on its intracellular region, and then the beta arrestin binds to the phosphorylated receptor, and the beta arrestin physically interferes with further coupling between the receptor and the G protein. This process is called desensitization because the receptor becomes less responsive even if the ligand is still present. The third mechanism of the receptor is the intentionalization. Beta arrestin can help recruit proteins involved in the endocytosis, causing the receptor to become moved into the cell within a vesicle. Once internalized, the receptor may be dephosphorylated and recycled back into the plasma membrane, or it may be sent to a lysosome for digestion. Internalization is not always equivalent to complete signal termination. In some contexts, beta arrestin and internalized GPCRs can initiate alternative signaling pathways. However, for the purpose of the overview of this major idea, that phosphorylation, beta arrestin binding, and internalization reduce conventional G protein signaling, and so second messengers are also removed. And then together, these mechanisms prevent overstimulation and control signal duration and allow the cell to then reset itself. So now we'll move on to clinical significances of while we're learning this. The biochemical importance of G proteins becomes specifically clear when we consider medicine. Approximately 1/3 of approved drugs have historically been estimated to act directly or indirectly on GPCR related targets. The exact percentage can vary depending on how targets and drug actions are classified, but an overall message is clear that GPCRs are among the most successful drug target families. Several familiar medications classes worked through GPCRs, beta blockers, beta androgen receptors, and they are used in conditions such as hypertension, certain arrhythmias, heart failure, and asthma. Or I'm sorry, not asthma, enigmas. Opioid medications act on opioid receptors, which are GPCRs involved in pain perception and other nervous system functions. Why are GPCRs such useful drug targets? First, they are accessible at the cell surface. A drug does not necessarily need to cross the plasma membrane to bind receptors. Second, different GPCR subtypes have distinct ligand binding pockets, allowing for researchers to then design these drugs with a degree of selectivity. Third, modulating a receptor can influence an entire signaling pathway and produce a meaningful physiological effect. Modern drug discovery also explore concepts such as partial agonism, inverse agonism, allosteric modulation, and biosignaling. Biosignaling refers to the possibility that a ligand may favor one receptor over another, for example, favoring G protein signaling or beta arrestin associated signaling. This may allow future drugs to preserve, the desired therapeutic effects while reducing adverse effects. So some examples of these are of the diseases areas and where GPCR signaling pathways is an important role. The first is cardiovascular disease, where GPCRs regulate heart rate, cardiac contracticity, blood vessel tone, kidney function, fluid balance, things like that. The second example would be in cancer, where mutations or abnormal expressions involving some GPCRs, G proteins, or downstream signaling components can promote excessive cell proliferation. The third is in asthma. GPCR influence contraction and relaxation of our airways, the smooth muscles, and so production and inflammation could cause you to be asthmatic. So beta 2 androgenic receptor agents are widely used for our bronchial tubes because activating these receptors promotes airway relaxation. Other GPCR related pathways can contribute to the bronchioles and inflammatory responses to be reduced, so then it'll make it easier for us to breathe. And then the next is you see it in diabetics. The fourth example would be GPCR signaling participates in the insulin selection, glucagon secretion, and appetite regulation. We see this a lot in the GLP-1 with these diet medications that have become very popular lately. And so here are the key takeaways that we should be focusing on, which is the signal amplification, how things are tightly regulated, and some of the major drug targets that we use that are affected by these GPCRs. And so in conclusion, I would like to emphasize these key ideas. First, GPCR signaling, activate multiple G proteins. Each effector can generate many second messengers, and each kinase can modify many target proteins. This allows the cells to respond to strongly to a relatively small extracellular signal. Second, the pathway is tightly regulated. GTP hydrolysis receptor phosphorylation, beta arrestin binding internalization, second messenger degradation, and protein dephosphorylation all help control the strength and duration of our responses. Regulation is essential because excessive or prolonged signaling can disrupt normal physiology. And third, GPCRs are major drug targets. Their accessibility, diversity, tissue distribution, and control over important physiological pathways make them central to our modern pharmacology and what a lot of drug developers try to target. The overall message is that GPCR converts extracellular signals into precise intracellular responses Their 7 transmembrane structure allows them to detect ligands outside of the cell, activate G proteins inside the cell, generate second messengers, and coordinate changes in metabolism through things like gene expression, secretion, and contraction. Understanding this pathway will give us the foundation for understanding normal physiology, disease mechanisms, and the action of many commonly used medications. Finally, here are some of the resources that were used in the development of this presentation. And so with that being said, I would like to thank you for your time and viewing my presentation.